Welcome to the lecture series on vitamins in nutrition. So, today we are going to look at the role of vitamin K and how vitamin K plays an extremely important role in terms of cell cycle. So, in the previous session we actually looked at vitamin K's role in atherosclerosis. So, that is one way by which we could always say how vitamin K plays an important role in pathology, especially when it is going to go out of control, and in that line we were looking at Gas6. So, this is one particular protein we need to be aware of that has a very close interaction in vitamin K related cellular mechanism. Now, if you look at Gas6, we have to have some important information regarding Gas6, and if we know its function, we would be able to relate how this particular protein can play an important role when they actually contribute in the atherosclerotic pathway. So, you look at this particular protein Gas6, which actually has or lacks a thrombin sensitive motif, as you could see over here. So, it has a sequence homology with epidermal growth factor. So, epidermal growth factor is generally considered an important, I would say, regulatory component in terms of cell proliferation. Okay, so, they lack thrombin sensitive motif. So, this makes them susceptible to thrombin cleavage. Suppose if you have this particular motif, the thrombin can go and cleave this particular protein and could initiate the process. But since they lack thrombin sensitive motif, their susceptibility to thrombin cleavage is now eliminated. Okay. So, now we go further. Now, this gas protein actually acts like a kind of a ligand for the reception of tyrosine kinase AX1 and SCY-RS2. So, these are some of the proteins that protects the cell from apoptosis by activating ARK phosphorylation and inducing MAP kinase activity. So, you may ask why we study all this. The reason is at the back, how vitamin K plays an important role in the thrombolytic and in the pathways connected with inflammation. Now, if you remember, vitamin K has been recently playing an important role in the anti-apoptotic pathway. Okay. So, you may ask a question, so whether apoptosis is really needed? Yes, apoptosis is important because that is the way by which our body says that old cells, please show your way for the new ones, so that you leave it without causing any inflammation. Now, this may not be the case for cells which are still active or in areas where you do not need regeneration or you need sustenance. In such cases, gas functions, and vitamin K is closely associated with those proteins which technically as gas related mechanisms. So, this is that ligand that actually goes over there and binding this particular pathway protects the cells from apoptosis. So, you see over there, so it has also been found to facilitate growth of smooth muscle cells. So, smooth muscle cells are an important entity in the vasculature. So, vasculature, constriction of the capillary walls and even the arterial walls are so closely associated with smooth muscle function. So, you see there vitamin K playing a role in the anti-apoptotic and also helps in the injury, and now we see over there how they indirectly play a role in the gas mechanism and thereby promoting vasculature. So, you may have, so it is something like kind of two sides of a coin. On one aspect, vitamin K plays a role in the initiating of clotting mechanism. On the other end, it also plays a role in kind of a cytoprotective environment. So, it tries to create a balance, and in any pathway you need to have a balance of either side. If that balance is not realized, then the pathway would go uncontrolled into a direction that can give rise to severe inflammation. So, you see there Gas6 may also function as a ligand for TYRO3 and thus serve as a neutrophic factor. So, neutrophic factor is basically a kind of a growth factor that helps in the proliferation of immune cells like neutrophils. So, now we are going to look at one more important pathway, how vitamin K plays a role in the sulfatide metabolism. So, you see there, this is basically an important pathway where sphingolipids are being produced by bacteria. Okay, so, you see there, this role would be very interesting because we are trying to derive certain components from gastrointestinal bacteria or the good ones that lay in a large intestine. So, vitamin K is not only being utilized by us, but it is being utilized by the bacteria which live in the gastrointestinal tract. This particular bacteria in the gastrointestinal tract has a very important role to play in terms of how they contribute in the sphingolipids. So, you see over there, they are supporting the activity of serine palmitoyl transferase. So, serine palmitoyl transferase, the initial enzyme in the sphingolipid pathway. See, we have this particular I would say lipids, very good lipids which are needed as raw material in the insulatory mechanism of the brain. So, you have plenty of sphingolipids in the brain because brain is a mass of, I would say, neurons covered by very good sphingomyelin and other phospholipids. You may wonder why we need to have this much amount of lipid in the brain. The reason is all these phospholipids not only provide an insulating environment, they also help in the normal growth and normal functioning of nerves. You should imagine this picture. So, you have a kind of electronic circuit. The electronic circuit, as you see, has the key components involved. I would say the circuit board, and then you have other accessory components which acts like a kind of an insulator. So, fundamentally sphingolipids along with other phospholipids in the brain play an essential role. Now, we are connecting the line. So, this sphingolipids produced by bacteria in the gastrointestinal tract would also contribute to the overall sphingolipid need of our brain. So, you see there vitamin K is like a kind of a added supplement to the sphingolipids. So, how vitamin K playing a role in the intestinal bacteria, and this in turn provides a raw material which is needed for the normal functioning or the neurologic physiologic responses of the brain. So, you see over there in the nervous system, thus the brain sphingolipid metabolism is extremely important in the brain. Okay, so, you see the sphingolipid components are not only involved directly in the insulation of the brain and the neuron and all the network of neurons present over there, but they also act like a kind of a second messenger system. So, we call it like IP3 DAG pathway. So, you have different pathways, I would say the cascade mechanisms. So, this I want you to be attentive. So, this is what we call it like the second messenger for intracellular signal transduction pathway. So, what do you mean by second messenger? So, you have a receptor and there the particular ligand goes and binds, and it is something like a kind of a ligand binding onto a switch. The moment switch is on, the circuit needs to complete, so that the final I would say component would be released that can probably migrate inside the cell and could go, I would say, across the cytoplasm to the nucleus and would result in the production of transcription factors, which in turn will govern the expression of important proteins that are needed in the normal functioning of the cell. So, you see over there, now picture all this that is happening inside the brain. Okay. So, all the neurons and the second messenger systems which are involved in the normal functioning of neuron and all those metabolic functions needed for the nourishment of neurons, okay, all being associated with sphingolipids. So, sphingolipids is basically the fundamental raw material, I would say the insulating material. You can also consider the myelin sheath. Okay. So, myelin sheath has its own range of phospholipids and other protective lipids being produced, and here you have sphingolipid being one of the raw material for majority of the lipids, or I would say the lipid environment of the nervous system. So, you look over there, this is very interesting to note, because how transcription factors are being regulated by vitamin K. So, what is this transcription factor? So, transcription factor is fundamentally, I would say, the key switches in the nucleus. Suppose if you have a DNA and there are stretches of, I would say, fragments of DNA sequences which produce, I would say, protein for the normal functioning of the cell. Now, these transcription factors are the ones which basically not only regulates gene expression, but also regulates the flow of activity in the cytoplasm of the cell. Now, what they have done in cell culture, they have taken like vitamin K1, K2, K3. Okay. So, when you add it over there, so they delay the cell cycle and enhance apoptosis. Okay. So, you remember previously we saw vitamin K through some of the mediator proteins have anti-apoptotic mechanism, but here we find that vitamin K can actually trigger apoptosis. Okay. So, it means what? Vitamin K would be there available when you need to wind up the cell activity and give way for new cells, and if suppose if in any case where there is a premature apoptosis related mechanism, vitamin K would have a say on those regulatory proteins which have anti-apoptotic pathway and through their influence. So, probably we could consider it a kind of a feedback mechanism that vitamin K is playing, but this information is a result of direct intervention of vitamin K on the apoptotic pathway. So, that is quite interesting to note. So, you see that vitamin K3 also appears to induce protein tyrosine kinase activation and to inhibit through direct interaction extracellular I would say signal regulated kinase, what we call it like ERK protein tyrosine phosphatases. So, you come across this word tyrosine phosphatases. So, what is a phosphatase? Phosphatase is something that could cleave the phosphate group, and you have ATP cleaved, you have ADP cleaved, you have AMP. So, at every stage you have a phosphatase cleaving the phosphate groups, and each of them in a very sequential manner have their own role to play in the second messaging I would say ability of the cell. So, you see over there these effects are associated with impaired proliferation. So, you see that. So, vitamin K when you add it to a cell line, okay, and if you closely monitor the cell cycle, so vitamin K appears to have a impairment on the proliferation. So, technically if you could take a fast growing cell line and you add vitamin K to it, you would eventually notice that the cell proliferation is coming down over a period of time, and this is the mechanism by which the cell proliferation is being brought low. So, if cell proliferation is brought low, then it means it is promoting apoptosis. This is very important because we are actually in the business of cancer therapy which could promote apoptosis. So, if you could have anything that would promote apoptosis for cells that are, I would say, deemed to undergo apoptosis, that is a good thing. Whereas in cases where there is premature functioning or dysfunction, where you want to inhibit apoptosis, that is also a good thing in terms of regulation, but those ones which are creating or the troublemakers, you want them to undergo voluntarily apoptosis. So, this feature of vitamin K is very interesting to note. So, you see over there they actually act as a cell cycle arrest. So, vitamin K3 has been shown to inhibit cyclin dependent kinases by binding to sulfhydryl at the active site of those enzymes. So, these are some of the, I would say, findings that tells you vitamin K's role in the direct inhibition of cell. So, you see that this effect is associated with inhibition of malignant cell proliferation at the G1S and SG2 phases of cell cycle. So, you should be like little familiar about this cell cycle business. So, the cell cycle is basically a kind of a cell cycle where you have two major phases. Okay. So, what we call it like gap 1 and gap 2. So, between the gap 0 and gap 1 you have the synthetic phase, and then you have the mitochondria, I would say division, cell proliferation phase. So, something like you see over there this is, if you take it as a cycle, this one can be considered as the G naught and this one as G1. So, G refers to gaps. So, what are the two major events, and this is that region what we call it like the synthetic phase, where all the raw material needed for cell cycle would be made, and M phase what we call it like the cells are actively dividing. Now, between the S phase and the M phase you have G naught and G1. So, where vitamin K is playing a role? Vitamin K is playing a role at G1 S and S G2. So, probably if you consider this to be the S phase, G1 and G2. Okay. So, that is what we have to look at. So, G1 and G2 are the two gaps that is very important for the cells to make things ready before they go into a major function or major action or activity. Okay. So, you see there cyclin dependent kinases. So, whenever you come across the word kinases, consider they are involved in proliferation. So, phosphatases cleave the phosphate group, kinases had a phosphate group, it is like phosphorylation. So, vitamin K3 is being found to bind with the sulfhydryl groups at the active site of those enzymes. So, sulfhydryl groups where you are likely to find in the cysteine residues. So, cysteine amino acid has this sulfhydryl group, and also you could exhibit or look at, I would say, methionine kind of amino acid derivatives. They also have a sulfhydryl group, because in methionine the sulfhydryl group is not completely free, probably it is a kind of methyl sulfur moiety attached, whereas in the cysteine the sulfhydryl group is very active. So, if you find these components being closely associated, so K3 can go and bind to the sulfhydryl group of cycling dependent kinases and thereby exhibit its effect. So, K2 has been known to affect cyclin function, which is also manifested as cell cycle inhibition. This is very interesting to note, because in the cell cycle inhibition you have a very good role to play in regulating cancer cells. Okay. So, you may have thought whether vitamin K can be used in cancer therapy. Yes, that is what we are trying to say now. So, vitamin K can be used as a adjuvant in cancer therapy. So, you see there in the oxidative stress of the malignant cells. Okay. So, I would say highly proliferating cancer cells would be very much susceptible to vitamin K, because for a normal cell compared to that, the cancer cells are into a mode of higher inflammation. Okay. So, what happens is vitamin K's ability to interact with the cell cycle dependent components, like what we call it like CDK, cycling dependent kinases, and vitamin K can directly introduce the phase of apoptosis, because cancer cells have defied apoptosis. So, they do not want to actually enter into the phase of apoptosis, because once it does, yes, the game is over, the cancer cells have to die. So, vitamin K would put cancer cells in a predicament where they are forced to take up apoptosis, because vitamin K has a direct impact on CDKs. So, this is a very interesting find, and this tells us how vitamin K, apart from its use, I would say as a dietary supplement and the nutritive role they play and all the good things that they contribute in the inflammatory pathway, for people who are adequately provided with vitamin K, can also have a kind of a protective measure in treatment of cancer therapies. Okay. So, you see over there, can vitamin K deficiency be pronounced? Yes, vitamin K, if at all, is not available. So, you say that deficiency in vitamin K can actually give rise to hemorrhagic diseases. So, what is this hemorrhage? So, hemorrhage is internal bleeding. So, as I have told you earlier, even in the beginning of vitamin K discussion, I told you we do not have any well established vitamin K deficiencies for an average healthy person, because most of the vitamin K is supplied from the dietary sources, but a prolonged deprivation can be a problem that could happen in patients who are not ambulatory, or probably who are on a restricted dietary mode and who have, I would say, been deprived of vitamin K for various reasons for a very long period of time, then the deficiencies can be really, I would say, pronounced. So, many of the popular rodenticides, as I have told you earlier, act by inducing vitamin K deficiency. In other words, when a person is being exposed to rodenticides, so you may consider, nobody is going to voluntarily take rodenticides until they are into a suicidal ideation, but rodenticide like compounds resulting as a byproduct of metabolites from variety of the chemicals that people consume as part of dietary supplements can actually be a culprit to induce vitamin K deficiency. So, what I am trying to say over here, vitamin K deficiency happening by itself in a natural way is very limited, but when people are being exposed to coumarin like rodenticide derivatives either directly or through some other sources can have a induced vitamin K deficiency. So, this kind of deficiency exposure for a prolonged period of time can lead to hemorrhagic diseases and eventually can end up in death by bleeding. So, that is the point we are trying to do. Probably in rodenticides we intentionally create a vitamin K deficiency, which is extremely unusual for rodents, because rodents diet is mixed. So, they somehow manage to have vitamin K, and taking or being exposing them to warfarin kind of components for a very long period of time can actually create a theoretical vitamin K deficiency in them, and this eventually would lead to the bleeding disorders. Okay, so, you see there coumarin derivatives are one of the important components, and there are some plants, I would say, that contain coumarin kind of compounds in them. So, we should be extremely careful when we consume them as food substances and make sure that they do not actually end up inducing vitamin K deficiency in the person who is consuming it. Okay, so, nowadays you have vitamin K, I would say warfarin kind of rodenticides available, which basically has a very long period of time. Okay. So, that a single feeding can kill not only the rodent, but can cause bleeding diseases in dogs and cats. Okay. So, it means what? This is a warning for us. Whenever we keep most of the rodenticides as a pest clearance, we should be very careful that they are not exposed, because we normally mix it with food and keep it. So, since it is able to create vitamin K deficiency in rodents, all mammals are susceptible. Okay. So, we should also make sure once a rodenticide is picked up by the particular poison, we make sure you completely clean the place, otherwise all the pet animals can also consume this, and this can induce vitamin K deficiency in them and eventually can end up in bleeding diseases, the hemorrhagic diseases. So, you may question whether this hemorrhagic diseases might be similar to that kind of a dengue related conditions. Yes, symptomatically it might resemble that, but this one is basically a vitamin K deficiency. So, how do we reverse it? So, you can reverse it by adequate doses of vitamin K, and this would actually create a kind of a higher dose of vitamin K, then overriding the mechanism, and thereby would help the enzyme produce sufficient amount of vitamin K that can be used as a cofactor to complete its normal metabolic role. Okay. So, you look over here, liver diseases, this is one particular case. So, here you find the liver synthesizes bile. So, I have been telling you time from the beginning in almost all fat soluble vitamins. So, if there is a problem in the liver, then that liver disease can actually be a precursor condition that can directly affect the dietary absorption of vitamin K. So, hope you get it, because liver is essential in producing bile from sterols available endogenously produced, and this can give rise to fat soluble vitamin absorption, and that is how a normal healthy liver would function. But in case with liver diseases like jaundice, hepatitis and all sorts of liver scarring diseases and cirrhosis, where I would say the output of bile salts may not be adequate enough to really enable appropriate fat soluble absorption. So, in such cases when this particular individual is deprived of fat soluble vitamins, vitamin K also comes under the list. Okay, so you see there we have to have an extra dosage of vitamin K from people who are suffering from liver diseases, or even for people who underwent liver transplantation where the liver would take little time to restore its normal function. So during that interim period we have to actually expose them to a wide variety of components, and within that period of time it is always advisable to supplement with vitamin D on a dietary source, or directly provide I would say vitamin D components through parenteral routes. Okay. Now you look at two other vitamin K deficiency, okay, so which is of a recent attention. So hemorrhagic disease of the newborn results from vitamin K deficiency in human infants. So this is of important notice because newborn children always have this problem of liver not being fully mature. So that is one of the reason you find they have jaundice symptoms. Okay, their liver takes time to kickstart. Most of the infants suffer from it, and it is like quite reversible. So within a week's time you expose them sufficiently and you give time for their livers to settle down, because the liver has to adapt. So far it was taking only the components available from the maternal source, and now the liver has to stand for itself and get itself exposed to wide variety of components. So most of the livers pick up, so you do not have an issue, but there could be a problem where I would say the hemorrhagic disease can happen because of the liver disease, and this eventually I would say leads to hemorrhagic diseases. So essentially there is no vitamin K. So since there is no vitamin K, so and the gastrointestinal tract is yet to be established, because the infant is still on I would say the diet of milk. So it takes time for the bacterial population to get established. So within this interim, I mean there could be vitamin K deficiency, and effects have been introduced, measures have been modified to include vitamin K as part of the dietary supplement. So otherwise if untreated, and especially if the child, if the infant has I would say long standing deficiency, that can give rise to a hemorrhagic disease condition. So this is one important point of attention that we need to consider. So you consider one more problem, two other vitamin K deficiency states that is of considerable attention would be I would say the fracture component. Although I have dealt with this earlier, I would like to emphasize how this is possible. So far our idea goes, we always associate D, calcium and parathormone, but now we see here vitamin K is also a culprit in this particular therapy. So it says that use of vitamin K antagonists like warfarin for coagulation therapies, anticoagulation therapies, these patients have to be closely monitored for bone density, because you are deliberately using a vitamin K antagonist, and thereby you might directly interfere in the physiologic role of vitamin K. So since vitamin K is also involved in the bone mineralization, these people are at high risk of fractures. So apart from the traditional view of looking at vitamin K as only a coagulant mediating vitamin, now these roles have come in the recent years, and all this states that vitamin K is such an important fat soluble vitamin, and we have this vitamin taking a key role in those areas which were unknown earlier. So as a quick summary, I would say vitamin K is one of the key fat soluble vitamin, although much attention has not been given to vitamin K as had been done earlier. So as a summary, I would like to say earlier all the fat soluble vitamins, especially vitamin A and D, had been given elaborate treatment including vitamin E, and vitamin K, because of its very little dietary deficiency conditions, have not been given much attention. But recently, considering these inherent I would say risks, especially for the population which are by birth like the infants and people who are on warfarin kind of therapy, would pose a special risk for vitamin K deficiency. So, that way vitamin K is such an important vitamin in the overall well-being of human physiology. Students can note down the important questions concerned with vitamin K metabolism. So, you can say what is the chemical structure of vitamin K and what are different types of vitamin K derivatives available and what is their role in human physiology. Then you can mention what would be the role of vitamin K in the coagulation pathway. Then you can also take down this particular question which says that what are the important vitamin K deficiency that you commonly find, especially for people who undergo therapy for I would say anticoagulants. So, I repeat the question, you have to know what are the classification of chemical classification of vitamin K. The second one would be what is the metabolic role of vitamin K, and the third one would be what are the specific deficiencies that people can have when they are on an anticoagulation therapy. Thank you.